Hello, dear students. In the previous video, we explored how kinetic proofreading helps aminoacyl-tRNA synthetases to reduce errors during amino acid selection. But here's the catch. Even after that checkpoint, some incorrect amino acids, especially those that are structurally very similar, can still slip through. Welcome to the double sieve mechanism, the second line of defense that ensures unmatched accuracy in protein synthesis. The enzyme doesn't rely on just one filter, it uses two. In this mechanism, which is called double sieve model, the first sieve blocks amino acids that are too large to fit in the active site. The second sieve removes those amino acids that are smaller but deceptively similar to the correct one. That means they are very similar to the correct amino acids. Together, these two sieves create an extraordinary level of specificity, ensuring that only the right amino acid is attached to its corresponding tRNA. So let us now explore the double sieve mechanism. The need for a quality control pathway to distinguish similar amino acids during protein synthesis was already predicted by Linus Pauling in 1950s. The molecular mechanism involved in such a process was discovered very later. The charging reaction, that is, the amino acylation reaction, itself proceeds in two stages. This we have discussed several times. First step involves activation of amino acid by ATP, leading to formation of amino acid and annihilate or amino acid AMP. In the second step, charging of amino acid at the CCA end of the cognate tRNA. This double safe mechanism says that there are two ways of editing. One, pre-transfer editing that occurs before amino acid is transferred to tRNA. Incorrect amino acyl AMP is hydrolyzed. The other part is post-transfer editing, means amino acid is attached to tRNA even after that if the enzyme finds that it has attached a wrong amino acid to a tRNA, the incorrect amino acyl tRNA is hydrolyzed. So we can edit both ways while activations, once the activation is over after that, and also after the transfer is over, then also we can edit. So after activation, before transfer to tRNA, editing is possible. After activation and after transfer to tRNA, then also editing is possible. Now let us understand this in much more detail. The double safe mechanism actually explains a size-based discrimination between the amino acids. The double safe model was proposed by Mikhail Frest and Heathner. According to this concept, there are separated active and editing site on the enzyme surface. The active site is first coarse sieve that attaches cognate isosteric or smaller amino acids while excludes larger amino acids. Okay, so what do we mean by this? The first site, which is known as the active site, will completely exclude the larger amino acid, but it will allow attachment of correct amino acid and smaller amino acids and also the isosteric amino acids. Okay. The editing site is the second site or the fine sieve that eliminates non-cognate mischarged amino acids through hydrolysis. Let us break down double sieve mechanism step by step using this diagram. On the left, you can see three types of amino acids. Correct amino acid, a near cognate amino acid which is very similar to the correct amino acid, and a third amino acid which is totally incorrect amino acid. Now all these three types of amino acid, when they approach aminoacyl-tRNA synthetase enzyme, the first checkpoint is the catalytic site, which is also known as the activation site, and this site acts as the first sieve. At this activation site, checking for the size and the structure of amino acid is done. Incorrect amino acids that are too large and very different from the correct amino acid, they are immediately discarded. However, near cognate amino acid, smaller amino acid, smaller than the correct amino acid, and the correct amino acids may pass through this first sieve. Once accepted, the amino acid is activated, and activated amino acid are then attached to the corresponding tRNA, forming aminoacyl-tRNAs. Now these tRNA, aminoacyl-tRNA, then comes to the second checkpoint or editing site. This editing site is also called hydrolytic site or the second sieve. At this second sieve, screening happens more vigorously. The site is more selective than the first site. Therefore, this is called the fine tuning site. It specifically recognizes tRNA that are charged with wrong amino acid. Such wrongly charged tRNA are directed to this editing site, where they are hydrolyzed and the free amino acid and the free tRNA are released from the editing site. But what happens to the aminoacyl-tRNA, which is the correct one? They do not fall to editing site and therefore they are not hydrolyzed. In summary, the first site removes large and incorrect amino acid. The second site removes amino acids that are small and deceptively similar. Together, these two sieves ensure that only correct aminoacyl-tRNA escapes from the aminoacyl-tRNA synthetase and are delivered for correct protein synthesis. This is the beauty of this double sieve mechanism. Let us move forward. Let us take this diagram to understand it in a better manner. This diagram beautifully illustrates the double sieve mechanism using amino acid size as the key factor. Now imagine this is your activation site on the surface of aminoacyl-tRNA synthetase specific for isoleucine. And supposing all these amino acids, tyrosine, phenylalanine, valine, alanine, glycine, and also the correct amino acid, isoleucine, approaches this enzyme. What happens immediately is the larger amino acids such as tyrosine and phenylalanine, they are rejected immediately from the first site, that is the activation site or the first sieve itself.
But this site allows or accepts the smaller near similar amino acid and the correct amino acid as well. So valine, alanine, glycine and isoleucine pass through the first sieve. When now they are activated and transferred to the tRNA. Supposing these aminoacyl-tRNA reaches the second sieve, that is the editing site. Here the smaller aminoacyl-tRNA will fall in at the second sieve or the editing site, which is also called the hydrolytic site, and the aminoacyl-tRNA will get hydrolyzed, releasing free smaller amino acid and free tRNA. However, the correct aminoacyl-tRNA, that is isoleucyl-tRNA, will never fall into the editing site and therefore is prevented from getting hydrolyzed, and they are released as a final product aminoacyl-tRNA. This isoleucyl-tRNA can now participate in the protein synthesis. So this is how this double sieve mechanism works. So error correction is done. From kinetic proofreading to the double sieve mechanism, you now understand how cells achieve near perfect accuracy. If this made things easy for you, hit the like button, share with your study gang and subscribe the channel, for more powerful concepts are coming your way. Keep learning smart and keep visiting Biochemistry Insights. Thank you for watching.